Hello and welcome to this revision video for Biology Paper 2. This is a video the night before the exam, so you've got your exam tomorrow morning. So make sure that you do keep healthy with a good night's sleep and a good breakfast, and do make sure that you've got all the equipment with you that you need. And remember, for biology, there's still a lot of maths in the paper, so be sure to bring your calculator to help you. I'm not going to cover all of the Biology Paper 2 content in this video. I do have longer videos that cover absolutely everything, and you can refer to those. I'm just going to focus your minds and pick out the key points that you need to know ready for tomorrow. First of all, then, we need to get into the mindset of what's going to be covered in the paper tomorrow, and this is topics 5, 6, and 7. So homeostasis and response, inheritance, variation, and evolution, and finally ecology. So you can forget all about your paper 1 stuff, your topics 1 to 4, and we are just revising and focusing on 5, 6, and 7. First of all, we'll discuss the required practicals. As you know, within the exam paper for science, you will get asked questions on the required practicals. Not all of them, perhaps one, perhaps a couple. For separate scientists only, if you do the hour and 45-minute exams, you will also have 2 more to do. But if you're doing combined science, there's actually only 2 required practicals for this paper. So they might ask you about reaction time. They might ask you about sampling organisms and their distribution. And if you're doing separate, they may ask you about decay or the plant growth required practicals as well. So that's where we'll start first of all. Let's look at this one here, the reaction time practical first of all. So one way to measure reaction time would be to use a computer game and get the computer to record the time it takes for you to respond to a certain stimulus on the screen, like there's a sheep one where the sheep are running across the screen and you have to stop the sheep at a certain point. Or you can do it practically using the ruler drop method. So you need to be aware of this one as well. So with the ruler drop method, someone holds the ruler just above your hand, so just in line with your thumb. And your thumb and first finger need to be slightly separated, and at a random point, the person will drop the ruler through your fingers and finger and your thumb, and you need to close your hand as quickly as you can, like this, and catch the ruler. And then from the top of your thumb, you can measure the distance. So if we were to look at this one, it was just below 15, so approximately 14 centimeters. Obviously, the longer the distance, the slower your reaction time, and you want a quick reaction time. So things that you need to be aware of, they might ask you about variables. So with this one, you could look at any independent variable that you wanted to. They might lead you to one in the exam, but for example, you could change the amount of coffee that you drank, the volume of coffee that you had. You could change perhaps the number of hours sleep that you had before you did the experiment. Whatever it is you want to change, you're looking at one factor and how that affects reaction time. So your dependent variable will be the distance it moves through your hand. And your control variables will be things such as the mass of the ruler, because if it's a heavier ruler, it might fall through quicker and therefore change the distance. So you want the same ruler because the mass needs to be the same. You would want to, each time that you do it, if you change the amount of coffee you're drinking, each time that you do it, you will need to use your same hand, whether that's left or right, but you need to use the same one in between each one. And you're probably going to want to have the same person dropping the ruler as well each time. The next required practical that you need to be aware of is organism distribution, and this is how we sample organisms. So they tend to be the organisms that can't move very far when we're talking about this method that I'm going to describe now. If we had a grassy area, we could then measure out a certain area within that with string let's say it's 10 meters by 10 meters by 10 meters. And then we would build our grid up, again using string. I'm not going to do this very neatly, but we would make a grid that is 10 by 10 by 10. It's not going to be very good, but 1, 2, 3, 4, 5, 6, 7, 8, and then that should make 9, 10. And then again the other way around. I'm not going to count them this time, I'm just going to do it roughly. But let's say we made a 10 by 10 grid. And so on. We need to randomly sample the organisms within that grid. That's the key for this experiment, random sampling. So you would measure out your string grid, and then you would give each square within that grid a number. So 1, 2, 3, 4, etc. so on until we got to 99, 100 down the bottom. Then we can't just throw the quadrat. Please don't write that in the exam. We need to use a random number generator. So that you can get these on phones, or you can get little tickers that do it as well. But we can get a random number generator, and that will pick us, let's say, 10 squares. So we'll put here, use the random number generator to pick 10 squares. So let's say, for example, that when we did that, it chose this one here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And we would then sample within those 10 random squares, and we would count the number of organisms that we find. So we could be looking at a plant, or we could be looking at some sort of animal that doesn't move within our study time. And we can then average those, and depending on the size of our quadrat, it might give us an average per half a meter squared or per meter squared. And then you might want to compare that to perhaps a shady area or an area where there is a different pH soil. You can test the pH using a pH meter, or test the light using a light meter, and so on. The other thing that you might want to do as far as sampling that you might see is using a transect. So let's say we have a tree. To sample away from one area to another, you would lay down a tape measure, and then every meter or 2 meters, but at regular intervals, you would place your quadrat. Didn't explain what a quadrat was up here. A quadrat is just a metal or a plastic grid. So this is the thing that we would then put on our square like this and count the number of organisms inside. So we can use a quadrat in both examples, random sampling and a transect. So here we would put our quadrat down at regular intervals, and we would want to count how many organisms are in our quadrat. Or simply you could just count or measure the organisms that actually touch the transect line. You can do a transect up a tree as well. For example, if you wanted to look at lichen cover, let's say this is lichen on the tree, you could do it up a tree by putting your tape measure vertically and at regular distances up the tree, regular intervals, measuring the size of the lichen as you go up, for example. So those are the 2 required practicals, nice and simple ones. So expect to either do a method or some calculations, or talk about those in your exam. Moving on to some content then. So some of the major content for topic 5, first of all. Well, topic 5 is called homeostasis and response. Homeostasis is all about maintaining a constant environment. So in the body, we want a certain temperature of 37 degrees C. In different parts of our body, we want to maintain an appropriate pH for the enzymes. So we need to make sure that we have controls in our body to do this. We're going to talk about the nerve system in response to this. So the nervous pathway that we need to remember is some sort of receptor cell detects a stimulus, which is a change in the environment. That will send a message to a sensory neuron, and that will send a message to the central nervous system, which is comprised of your brain and your spinal cord. So in most nerve responses, your brain is involved in coordinating the response, and that will go to a motor neuron. And finally, you finish with an effector, which is actually going to do something, and that's normally a muscle or a gland. So at the end of this pathway, you might cause a muscle to twitch and move out of the way. For example, if you just burnt yourself on a hot pan, you would have had pain receptors that send a message to your sensory neuron, to your brain motor neuron effector. But actually, when it's as quick as a burn from a pan, you wouldn't actually go through the central nervous system. So this is for most nervous pathways. However, my example there would have been for a reflex. So if I was to touch a hot pan and I had pain receptors in my hand that detected that I was in a little bit of trouble there because of the heat from the pan, actually it would avoid this part here. And rather than having a central nervous system, it would have a relay neuron in place of that, because this is an example of a reflex response when you're acting really quickly to get away from the danger. So in a reflex response, it is the relay neuron that is involved. So for separate scientists, you need to know about the muscles in the eye that are involved in the reflex response of bright light, which is all to do with the circular and the radial muscles. So normal nervous pathway involves the brain. A reflex does not go to the brain. It just goes, it zips across the spinal cord in a relay neuron and gets passed very, very quickly. At this point here, within a reflex pathway, when the message is being passed from neuron to neuron, so for example, when it's being passed from sensory neuron to the relay neuron, it has to go across a little gap, and that gap is called a synapse. So if I draw one neuron here, just finishing like so, and another neuron here, the synapse is the gap in the middle. So when the nervous signal gets sent along the neurons, that is an electrical signal. But at this point here, it turns into a chemical signal just so that they can get passed across that little gap there. So we're not sparking an electrical signal across a gap. And I'll write down here chemical. And on the other side again, that gets transferred into an electrical signal. As part of homeostasis and response as well, you need to know about the endocrine system, which is all the glands that are in your body. The master gland turns on and off various other glands in your body, and that is based in your head, and it is called the pituitary gland. The next one down is in your throat. It's responsible for metabolism, and it is the thyroid gland. Just by your kidneys, on top of your kidneys, you have the adrenal glands. It's where adrenaline is produced. And you've got a pancreas here, which you know produces lots of enzymes, but it also produces lots of hormones, which is what the endocrine system is all about. So the pancreas here as well. And finally, depending on whether you are a male or a female, you will have testes or ovaries. So testes are outside of the body and ovaries are inside of the body. And all of these glands produce hormones. Hormones are much slower than the nervous pathway. They might ask you to compare the two tomorrow in your exam. Well, the hormone is a much slower pathway. It travels in the blood. So hormone is a chemical messenger that travels in the blood. One specific hormone that we need to be aware of is in relation to the pancreas, which we'll talk about just now because it's a matter of life or death. In homeostasis, one of the things that's important for us to maintain is our blood glucose levels. And for foundation, you need to know the response that occurs when the blood glucose is too high. So this is the sugar that's in your blood. If it's too high, the pancreas releases a hormone called insulin into the blood. So you might have heard of insulin before and people injecting with insulin if they've got diabetes, but normally we produce it naturally via our pancreas. When that happens, the glucose moves into, or excess glucose that's in our blood, moves from the blood into our liver and our muscle cells. And then that glucose is converted, because it's soluble, we need to convert it, otherwise it will all move out again. It is converted into a substance called glycagon, which sounds very similar, glycagon, for storage. Okay, so if your blood glucose is too high, we want to maintain homeostasis, a constant internal environment. So our pancreas produces insulin, the glucose moves out of the blood and is converted into glycagon in the liver and muscle cells. Talking about the two types of diabetes that you need to know about, type 1 is the inherited diabetes, and type 2 diabetes is the one that can be brought about in later life because of a number of different risk factors, including poor diet and lack of exercise. Final thing then for topic 5 in relation to hormones really is the menstrual cycle. So that is the cycle that a woman will go through once a month. The main hormones involved are FSH, that's follicle stimulating hormone, that's produced in the pituitary gland. LH is luteinizing hormone, again produced in the pituitary gland. And you've got estrogen and progesterone that are produced in the ovaries. So the ones just with letters here, they are produced in the pituitary. And these ones here are produced in the ovaries. Now, FSH does what it says on the tin, follicle stimulating hormone. Follicle is the pre-egg stage. So before it becomes a mature egg, we call it a follicle. So the follicle stimulating hormone causes the egg to mature. The luteinizing hormone causes the egg to release, and that is on day 14. The estrogen, for higher I'll talk about later what that does in terms of switching on enough other hormones as well. But the main thing it does, it causes the uterus lining to grow, and the progesterone maintains the uterus lining. So if we were to have a look at what the uterus lining looked like, at the beginning of, let me just rub that out, and I'll just draw it under here. The uterus lining, first of all, at the start of a woman's menstrual cycle, she will have her period. And the period is when the uterus lining breaks down. So we're starting off thick and it's breaking down. And that lasts around 4 days. Then the uterus lining will start to build up again and then will be maintained until again she will have a period every 28 days. Now at this point here, this is when the egg is released. At day 14, the egg is released. It then hopefully attaches onto the uterus lining, and if it's fertilized and developed correctly, it will develop into a baby. So that's topic 5. A lot of different names to learn, a lot of different parts of the body, all about how hormones and nerve responses occur in the body. Topic 6 then, inheritance, variation and evolution. Let's go through the main parts that we get appearing in topic 6. We need the idea that all of our genetic information is stored in a molecule called DNA, and we need to be aware that this molecule is that double helix shape molecule. DNA, a length of DNA is called a gene, and that will code for a particular characteristic in your body. And the genes can be packaged, when necessary, into chromosomes. All of your DNA in your body, all of your genetic information is called your genome. And you would have heard of the Genome Project, where the scientists have studied all of the DNA in various different animals, including humans. So this links into reproduction as well as part of this topic. So the basics of DNA, and this links into reproduction because there's two different types. You can have sexual reproduction. That's when you have sex cells called gametes. For example, you could have a sperm and an egg cell, or a pollen and an egg cell in terms of a plant. In this case, we've got a sperm and an egg cell. These are our gametes, our sex cells. When these combine at a stage called fertilization, you will then have DNA from the father, DNA from the mother, combining to produce variation. So this method here produces variation. You're going to get a completely different looking child. It's not going to be identical to the parents. The other type of reproduction that you get is asexual reproduction. And in asexual reproduction, you are producing clones. So, for example, if this was the parent cell, this would simply divide and make two identical cells. And some larger animals like starfish and things like that can do this process too. A lot of single cellular animals, but some larger ones too. So sexual reproduction is the one that produces variation, and asexual reproduction is the one that produces clones and identical organisms. You will see some genetic crosses in this topic. For example, you can prove why we get a 50-50 male to female ratio, because the female eggs carry a special chromosome called the X chromosome. So that would be in the eggs, the 23rd chromosome. In our body, we have 23 pairs of chromosomes in every cell. So that's 46 chromosomes, one that you get from your mother and one you get from your father. So the 23rd pair is called the sex chromosomes. Females can only ever have an X chromosome. They are called XX. And males are called XY for their sex chromosome. So you might be asked to complete a genetic cross like this. All you do is you track along like this, and that brings an X from here and X from there, so that will make XX. This one over here, again, X and X, that makes XX. This one here would be XY, because we've got a Y here and an X here. We normally put the X first rather than saying YX. And finally, this one over here would again be XY. So if we do this cross, we prove that 50% of the offspring are going to be female and 50% of the offspring are going to be male. So we would indicate that somewhere on our diagram, 50% female, like so. And we would write the same for male down here. Things get a little bit more complicated in these genetic crosses. For example, let's say we had two different alleles. Alleles are a version of a gene or a type of a gene. For example, if we're talking about the colour of fur on an animal, you could have capital R to mean red fur and little r to mean white first. So our two different alleles here. When we're talking about the letters and the genetics, we're talking about genotypes. When we're talking about the words and what they look like, we're talking about phenotypes. So when we do the crosses, we can have different ones. For example, if you're doing foundation, they will probably fill in the alleles for you, like so. And all they will ask you to do is complete the cross. So, for example, in this case, we've got a homozygous dominant, because if it's a capital letter, it's dominant. And we've got a heterozygous, which means one of each, one capital, one lowercase. So if we cross these, just like before, we'd get R, R, capital R, capital R. In this case, capital R, little r, and capital R, little r. So to be white, it would have to have both recessive alleles, RR, but to be red, it could have a combination of RR or big R, little r. So in this case here, all of our genotypes has a capital R in it. If it has a capital, that colour will be expressed. So in this case, we have 100% red offspring. We could do another one though, whereby perhaps we have homozygous dominant and homozygous recessive, and then that would make 4, 1, 2, heterozygous, so a different cross, but it still makes 4 heterozygous and therefore 4 red offspring. And then the only other one, a couple more that you might see, we'll just do one though. If you had heterozygous for both, you'd have capital R, capital R, capital R, little r, capital R, little r, little r, little r. So in this case, we would now have 25% of our offspring being white and 75% being red. Because this one doesn't have any capital letters, so that means it will have white fur. If you're doing higher tier, this is one thing for extra for higher tier, and that is that you will actually have to build your own genetic crosses. So they will give you a load of information about whether the people or animals involved are heterozygous or homozygous dominant or recessive or whatever they are, and you will have to, just like I did, build your own genetic crosses. If you're doing foundation, you'll be given these crosses and expected to fill them in, so it's nice and easy, just tracking along with the right letters. Still then, for topic 6, a couple of extra things to go through. So there's a couple of key conditions that you need to be aware of. There's one that affects the mucous membranes in the lungs, and this is called cystic fibrosis. This disorder is caused by recessive alleles. You need to know that. So what this means is the offspring will only be born with cystic fibrosis if they receive the genotype that is homozygous recessive. If they were to get anything else, like so, they would not get cystic fibrosis. You only get that if you have both recessive alleles. Polydactyly is another disorder where you are born with more digits than you should have. And with polydactyly, that is actually dominant. So with polydactyly, even if you have big R, little r, or big R, you will have the condition where you're born with too many fingers or toes. However, even though it's dominant, this allele is rare in the population. That's why not many people are actually born with extra fingers and toes. It is quite rare, but it is a dominant allele. A bit of classification now. So king for kingdom, phylum, class, order, family, genus, species. This is the way that we classify and group all of our animals. This bottom section is really important for how we name animals in what we call a binomial system. So owls, let's take an example of a barn owl, for example. A barn owl might be called different things around the world within different local places. However, scientifically, it is always called a Tyto alba. This is the binomial system where they're given two names. So this name is the genus part, and this name is the species. So they might give you an animal, so they might say the tawny owl, Strix aluco is, and then they might say what is the genus of this animal or what is the species of this animal. So in this case, if they ask for the genus, that is the Strix part, and if they ask for the species, that is the aluco part. Okay, take a break if you need to, but if not, we'll move straight on to ecology. A couple of new key terms that they've put in. First of all, this picture here shows a group of animals of one type, of one species. And in this sense, we call it a population. So if it's just one species, a group of one species, it's a population. In this picture, though, we've got birds, we've got bears, we've got moose, we've got deer, we've got foxes, we've got snails, we've got mushrooms. So because we've got different species, we would call this one a community. So animals and plants are affected by several different factors. It's how they choose where they live in certain cases, or it's how they affect how well they do survive in certain places. And these factors can either be abiotic, which are the non-living factors, and biotic is living factors. So if you remember, biology is the study of living things, then that will help you remember that the bio on its own is the living factors, and the abiotic is the non-living factors. So non-living tends to be things associated with weather, such as temperature, rainfall, maybe things like pH in certain situations as well. Biotic are living factors, so the food availability, the competition that animals or plants are experiencing from other organisms, and predators and disease or pathogens as well. Really common question is for an examiner to ask you how animals or plants are adapted. And there are three different main types of adaptations that you need to be aware of. Those are structural adaptations. That's what the animal or plant actually looks like. There are behavioural adaptations, which is how do they behave? For example, do they live a solitary life on their own, or do they go around in groups? How do they behave with each other and with other animals? What are they dependent on? Things like that. And also functional adaptations, which is anything to do with inside the body and processes such as metabolism, temperature regulation, reproduction, and anything that happens inside the body that might be different between different organisms and help them give particular advantages for survival in different situations. Linking in with this ecology topic as well, then we go through our food chain. So our food chain always starts with a plant or an algae. So this is our producer that's producing its own food by photosynthesis. That is eaten, and the energy goes this way to a primary consumer, Primary consumers are eaten by secondary consumers. And finally, you may well see and have to use the word tertiary consumer, which is the third animal along in the food chain. For example, you could have your plant as grass, and your primary consumer might be a rabbit, and your secondary consumer might be a fox, and your tertiary consumer may well be an eagle, and you would show the energy being transferred as you go up your food chain. So, as I said, I haven't covered everything in this video, but I've just given a snapshot of some of the things that are coming up. The final couple of things for foundation that I want to go through are the water cycle and the carbon cycle, and then everything after that will be for higher and separate scientists only. Excuse me. So, final thing for combined foundation scientists are these two cycles that you need to be aware of. This one here is the water cycle, and this one here, as it says, is the carbon cycle. Now, water, H2O, can either be a liquid, a gas, or a solid. So in this cycle, it's really the key words that you need to be aware of. Probably a 3 or 4 mark question. If we start with water in the sea, that water can be evaporated and that will rise up into the atmosphere. You then get condensation where it turns into clouds, and precipitation when you get the rainfall and the water leaving the clouds. And then finally you can get flow where the water flows back through the rivers and streams and back into the sea. One final, so the evaporation doesn't have to be from the sea, it can be from the land or the rivers as well. One final part of the water cycle is linking into plants and topic 4, which is when plants take up water through their roots, it travels up their xylem, out of their leaves in a process called transpiration. So lots of keywords there, but they have parallels with physics as well, evaporation and condensation, so that one should be pretty straightforward. The carbon cycle is how carbon is cycled around. So carbon can be found in many different molecules, for example, CO2 for carbon dioxide, CH4 for methane, C6H12O6 for glucose, and here it's trapped in different molecules and it can be moved around the planet and Earth in a variety of different ways. So first of all, carbon dioxide can be taken in by plants through photosynthesis. They will then respire and they will release CO2 back into the atmosphere. The animals will eat the plants, so they're eating the glucose that they've made in photosynthesis, and they're transporting that through their body and using that for a number of different things within their body. You get microorganisms that respire when they decay waste materials, and underground as well when they're decaying dead and rotting things. The animals are respiring as well, and they are releasing CO2. And a couple of man-made things as well that are integrated with this. So mainly emissions and burning things. When we burn woods and carbon-based fuels like coal, we will also be releasing CO2 and cycling it back into the atmosphere. Obviously, with biology, this links into global warming. What we want is for not very much carbon dioxide in the atmosphere, because that is a greenhouse gas that can help heat up the Earth. And we want lots of trees to be able to trap the carbon dioxide through the process of photosynthesis. So it's a really important balance that we are facing at the moment to maintain low levels of carbon within the atmosphere. So if you are a combined foundation scientist, very best of luck for tomorrow. I hope the exam goes well. Don't forget to take all your equipment with you, and I hope it goes well for you tomorrow. if you are a higher combined scientist, there's a few extra things that you need to know. As we've seen in the paper 1 papers, it's quite important to hone down these few higher tier only contents, because it's quite likely that they will ask you some of this tier, otherwise yours might be too similar to the foundation paper. So we've got glucagon, hormone level interactions, adrenaline and thyroxine, IVF, genetic engineering, and we've talked about this one before, whereby you will actually need to construct a Punnett cross, a genetic cross from scratch. Starting off with glucagon then, you need to know what happens when the blood glucose is too high. We've said that before. We said that the pancreas produces insulin, and the insulin causes the glucose to move into the liver and muscle cells where it's stored as glycogen. For higher tier, you need to know also what happens when it's too low, and unfortunately this releases another word to us, which sounds very much like glucose and glycogen. So this one is called glucagon. So if the blood levels are too low, again, it's our pancreas involved. Our pancreas releases a hormone called glucagon. And glucagon helps us to get our glucose back from where we stored it. So it makes the liver turn the glycogen back into glucose, and then the glucose is then released. I'll put down here, glucose released into the blood. So if you're doing higher tier, you must know this pathway as well. You may well be asked a 4-marker linking the two things. The next thing that you need to be aware of in a bit more detail is the hormones involved in the menstrual cycle and how they interact with each other. So you may well have seen it in practice questions whereby you have 0 to 28 days or 1 to 28 days, and you have different levels of hormones here doing different things at different times. You might have a peak in one and so on, and then different things happening. You need to be able to work out what's happening and why. And it's all to do with how they interact with each other. So let's start off at the pituitary gland. Here we have FSH being produced. FSH stimulates the egg to mature. So this has to travel through the blood to the ovaries. So here are the ovaries down here and here. So the FSH travels through the blood to the ovaries. And when it's there, it stimulates the egg to mature, but it also stimulates the ovaries to start producing estrogen. That estrogen then travels to the pituitary gland, and it at the uterus lining, it will start to build up the uterus lining, but it also interacts with the other chemicals as well. Because what it does, we said that FSH stimulates estrogen production. Well, estrogen goes back and tries to stop or inhibits the FSH, and instead it stimulates the release of LH. So at this point, the estrogen has gone up to the pituitary gland, and then we get another hormone being released, which is our LH, our luteinizing hormone, and that causes the egg to be released. When progesterone is also produced by the ovaries, the progesterone helps to maintain the uterus lining. So the progesterone also wants to inhibit the FSH and LH because it doesn't want any more eggs to be matured and it doesn't want any more eggs to be released. So these two here are the ones that use in contraceptive pills, because they are the ones that are inhibiting the hormones which stimulate the egg production, and stimulate the egg released. So if we try and stop that by putting that into our contraceptive pills, hopefully we're going to prevent us getting pregnant. But that's how they interact. You need to know that in detail as well. IVF is a topic that you need to know in detail. For higher tier, you need to know that IVF is in vitro fertilization. And what this means is that fertilization, rather than taking place naturally inside a woman after the point of sexual intercourse, this is fertilization that actually happens by a scientist in a lab. So what they will do, they'll take a sperm sample from the male and they will pick the most motile sperm, the most active sperm, and they will take a number of eggs from a woman. Now, the process to get these eggs is quite difficult because the woman has to inject herself with FSH and LH, because normally once every 28 days she will just release one egg, and if you did that, the process of IVF would take years and years and years. So if she injects herself with these two hormones, it will help her, first of all, mature lots of eggs, and secondly, release lots of eggs. So that's quite an emotional and a difficult time for the woman to do that. But when we've got all these eggs released, then in a petri dish or dish of some kind, you'll have the woman's eggs in there. And again, the scientist will pick the best ones. He will then inject, or she will then inject, a sperm into each of the eggs. And then they'll leave that for a few days and hopefully they'll start dividing into the very early stages of an embryo. And they will look to make sure that that embryo looks really healthy. Embryo. And at that point, they will implant it into the woman's uterus. Now, at the point where IVF wasn't very successful, they may well have put up to 8 or so embryo back into the woman's uterus. However, because it's getting more and more successful, they normally only put around 2 embryos, as far as I know, back into the woman, and hopefully one of those will be viable. So often IVF can lead to twins because both embryos are viable embryos and can develop into babies. Now, you need to know the emotional stress that this causes the women in relation to IVF, and you may be asked to evaluate, so give you advantages, disadvantages, and your own conclusion. 2 hormones now that you need to be aware of for higher tier: adrenaline and thyroxine. Adrenaline is produced, as it's hidden in the name, in the adrenal glands. And this is responsible for our response to a situation that we call fight or flight. If you're suddenly under a lot of stress and you have to, a lot of immediate stress, and you have to, for your own safety, respond very quickly, then this hormone allows us to do that. So, for example, if you were standing in front of a lion, you would release adrenaline, and either you would exhibit a response of fight or flight. So you'd either try and fight the lion or you try and run away. And to do that, you need your muscles to be working to the best of their ability and your brain to be thinking fast and working to the best of its ability. So you kind of need to know this one off by heart, really. The adrenaline is released into the blood, and you need to know that that's from the adrenal glands. And you need to know that this increases the supply of oxygen and glucose to the brain and the muscles. Okay, that bit is absolutely vital. But obviously, if you're talking about oxygen and glucose, you're linking into the ability to respire more quickly and efficiently in response to the fight or flight situation that you found yourself in. So respire, maybe not more quickly, but really efficiently is probably better. So thyroxine then, this is produced in the thyroid gland, and this thyroxine helps to control your basal metabolic rate, which is your metabolism. It's the basic metabolic rate that you need whilst at rest. The release of thyroxine from the thyroid gland is controlled by another hormone called TSH, which is thyroid stimulating hormone. And you may well see the term negative feedback in a number of different places in this biology course. A negative feedback means if the levels of something in the body are too high or too low, the body will respond to counteract that. For example, if we kind of sketch out our thyroxine levels, if this is maintaining our basic metabolic rate, we wouldn't really want it to go up and down like this. We would want it as much as possible to stay at this optimum level. However, they will go up and down but they are controlled by negative feedback. So thyroid stimulating hormone, TSH, would stimulate the thyroid to produce more thyroxine. So at this point here, when the thyroxine level is increasing, actually the TSH needs to be inhibited. Okay, so they're not producing as much TSH and the thyroxine levels go down again, but due to negative feedback, is not allowed to drop completely. And at this point here, you will have TSH stimulated and released so that it then brings back up the levels of thyroxine. So if it gets too high, something happens to bring it down again. If it gets too low, something happens to bring it up again. The final piece of content that you need to know if you are a combined higher scientist is genetic engineering. Genetic engineering is when you take a gene out of one organism and you put it into another. So if you do this in plants, you are producing a GM crop, a genetically modified crop. For example, you may want to take the gene for drought resistance. Let's say this is the drought resistance gene. And you might want to take that length from the other long strand of DNA that you've got, and you might want to put it into the DNA of a crop that you want to grow. For example, you might want to put it in your wheat crop. So what you would do is you'd use enzymes to cut the gene out of the plant. For example, if we wanted drought resistance, we'd find a drought resistant plant, we'd use enzymes to cut the gene, and then we would put that into the genetic information, the DNA of our wheat plant. So you're taking it out of one plant and you would literally, again, cut the wheat DNA and stick it in there. So with our drought resistant gene, we would then stick it in there and we would have our genetically modified crop. We can do this for bacteria, so we can have, for example, the gene for producing insulin. We can cut this from where it was in our human DNA, and then we can insert it into a bacterial plasmid and we can put it in here, and then we can put the plasmids back into a bacteria, and the bacteria is therefore then going to produce insulin from us. So we've genetically engineered it. We've taken the gene for insulin production out of the human, and we've inserted it into the bacterial loop, the plasmid, the bacterial DNA, sorry, the plasmid of the bacteria, and then we can put those plasmids back into the bacteria. And that bacteria will rapidly multiply again and again and again until we had loads more. and all of them would be producing insulin because of the plasmids that they transferred on. So if you are doing higher tier combined, that is the end of the video. All the best for your exam tomorrow. I really do hope it goes well. And if there's anything else that you need to ask, then you can ask a question on the frog wall. Separate scientists then. I'm not going to do a long addition to the video at the end for separate scientists. What I'm going to do is highlight the bits that are separate science only, and I will refer you to the video that I uploaded a few days ago that has all of the content that you need. Because at this point, you probably don't want to go through everything. There might just be a couple of topics that you need. But we will focus first on the required practicals, and I will go through those for you in this video. So for separate scientists, as well as random sampling organisms using quadrats and transects, and the reaction time combined practicals, you will also need to be aware of a decay required practical and a plant growth required practical. What I want to emphasize for paper 2 is how full this screen is, and therefore how much content there is for separate science only. So in paper 1, there's only a few additional bits that you could kind of guarantee were going to come up for separate scientists. But in this case, we've got the brain, we've got the eye, sexual and asexual reproduction, cloning, temperature regulation, water control, protein synthesis, DNA, plant hormones, work on famous scientists, food security and production, and trophic levels. Within these, there are just a few little snippets that are higher tier only, which I'm going to emphasize in this video. And there are a couple of complete little sections that are higher tier only as well. So if you want to find out information on any of these in detail, then please do look at my previous video. But for the night before the exam video, we're going to go through the required practicals, and then I'm just going to highlight the higher tier only content for you. So the decay required practical. Decay is carried out by microorganisms. So microorganisms, let's just draw one here. This is our microorganism. If that wanted to decay rotting matter, what it would do is it would release enzymes into the leaf litter or the rotting animal, whatever it is, outside. Those enzymes would break down the longer molecules in the decaying matter, and then the soluble parts, the soluble molecules, can move back in. So for example, this could be, take a extreme example, they could secrete amylase. Amylase could break down starch into glucose, and then glucose is a soluble molecule that it can then take up again here. Oh, sorry, I've gone on to the other slide. So this is decay. So that is how decay works. So you need to know information about that, which I'm not going into all the detail here, but the microorganisms are generally respiring through decay, so they need oxygen, and you need to know about biogas generators and anaerobic decay, but that's in my other video. Let's concentrate on the required practical then for decay. First of all, we're going to use the enzyme lipase, or they may give you an example of any enzyme in the exam. So they might be talking about something else in a completely different situation. Let's write that again clearer. So we've got lipase. Lipase breaks down lipids, and we find lipids in substances such as milk. What we're going to do is see how temperature affects the rate of decay. So what we need is a known volume of lipase that we've measured out using a pipette or a measuring cylinder, and the same of milk. We need to add an indicator, and for this one we'll use phenolphthalein, and we would put, say, 5 drops in there, and we would also add some sodium carbonate. the sodium carbonate is going to make our solution an alkaline solution. So we would put these into test tubes or boiling tubes. And if we want to test how temperature affects the rate of decay, what we would do is we would put these in a water bath, and our independent variable would be the different temperatures of water. So we might have, let's just write down temperature of the, not temperature of the, well, it's the temperature of the water, but it's the temperature of the enzymes and milk, or you could just put temperature of the reaction. So we've got our water bath here. We could set it at 30 degrees C, for example, as our first temperature, and then we could change it to 40 degrees C, 50 degrees C for other measurements in the experiment. We've got our lipase in our water bath and our mixture here of milk, indicator, and sodium carbonate. Now, first of all, this will go a pink colour because of the sodium carbonate and the fact that it's an alkali. So this phenolphthalein will go pink, and when the pH lowers to a certain endpoint, it will go colourless. So we make sure we put our rod into stir and make sure it's even. And what we'll do is we'll take a small amount of lipase, say 1 centimeters cubed. We will add that to our mixture and stir, and then we will time, which is going to be our dependent variable, time how long it takes to turn colourless. And at this point, we know that our enzyme has decayed the lipids that are in the milk. And then we can change our temperature, repeat it again, and so on and so forth. So because we're not measuring a certain thing for rate, we're just measuring the time it takes to change colour, this is where that funny equation, rate equals 1,000 divided by time, comes in, which gives you a rate per second of decay. So you may well be asked to write a method for that practical, talk about particular volumes, never talk about amounts. Talk about volumes of lipase, milk, sodium carbonate, and don't forget your indicator in there as well. Changing the temperature and timing how long it takes to go colourless. The other required practical for separate only is a relatively simple one. What you need to do is talk about how something such as light or gravity, they'll let you know in the question, affects the plant growth. So, for example, you could have a Petri dish and you could put a certain number of seeds on that. You could then put a light in this position and then record the growth and draw diagrams of the growth of the plant. So you would want to, after a little while, you'd have plants growing out of here, and they might be bending slightly towards the light. One should be bent a little bit more that way. And you would need to draw detailed drawings of what's going on and accurately measure the lengths of the seedlings that you are drawing. You may then want to change the position of the lamp so it's not over here anymore. And instead you might want to put it on the other side, like so. And again, do exactly the same thing. Draw, record, measure the height. If you're measuring the height, you need to be clear about how you're measuring it. You might want to measure it vertically upwards like that, rather than that way. But if you're measuring the length of it, you might want to measure the full thing like so. So that's the plant one. So you would be taking detailed drawings of how the plants are growing. If you had gravity, rather than light, you might put the Petri dishes on their side and on the bottom on the other side, and you're going to be measuring and recording growth that way. So the plants would obviously grow upwards and so on and so forth. So it's all about setting up an experiment and making sure that you're accurate with your drawings and your measurements. So the things that you are changing in this one would be the position of the light. Things that would be changing in this one would be the effect, the position of the Petri dish, sorry, and then you are measuring the length of the plants or the direction they're going in or whatever you are asked to do. So for foundation separate scientists, that's where you can now leave. So all the best for tomorrow's exam, and I really do hope it goes well. For separate higher scientists, I just want to go through the extra few bits of higher tier only content. I'm just going to go through them briefly just so that you are aware of them and you can access them on my other video if you need to. Why I do this is because you're doing separate higher, you might want to hone in on this extra little bit of the content, although there is a lot of separate content only, so they might ask you absolutely anything when it comes to tomorrow. Higher tier only then, a bit about the brain. You need to know a little bit about these scientists, neuroscientists that work on the brain, and you need to know that they've been able to map regions of the brain to particular functions by studying patients with brain damage, electrically stimulating different parts of the brain and using MRI scanning techniques. But the complexity and delicacy of the brain makes investigating and treating brain disorders very difficult. So you may be asked to evaluate the benefits and risks of procedures carried out on the brain and nervous system. Evaluate is where you give your advantages and disadvantages, and then you must finish with a justified conclusion at the end. For a bit about temperature regulation, in your content you need to talk about vasodilation and vasoconstriction, and as well as that, sweating and hair standing up on end and so on and so forth. Just be aware for higher tier, you should explain how these mechanisms for temperature regulation lower and raise the body temperature in a particular given context. So just be aware they might give you completely random context, but just read through it carefully and think about what mechanisms are going to be involved in regulating body temperature. For water control, so linking into the kidneys, if you're doing higher tier, you need to describe the effect of ADH on the permeability of the kidney tubules. That's how much water is reabsorbed back into the blood. So the water level in the body is controlled by the hormone ADH, antidiuretic hormone, which acts on the kidney tubules. ADH is released by the pituitary gland when the blood is too concentrated. That means it needs more water. It causes more water to be reabsorbed back into the blood from the kidney tubules. And this again is another example of a negative feedback system. The other thing that's particularly higher tier is this idea of deamination. So when excess amino acids are deaminated, this means digestion of proteins from the diet results in excess amino acids, which need to be excreted safely. In the liver, these amino acids are deaminated to form ammonia. Basically, that just means the amino acid has its amine group taken off. Well, you don't need to know about that, but the nitrogen side of it taken off, and it's turned into ammonia. So basically, from the amino acid, deamination gives us ammonia. Okay, that's what deamination is, turning the ammonia into amino acid and another byproduct that you don't need to know about. So the ammonia is toxic. That's bad. That's going to be bad for our bodies. So it's immediately converted into urea for safe excretion, and then that links into the production of urine. Okay, so we know already proteins make amino acids, but just this extra step, deamination, is when those amino acids when ammonia, is made from those amino acids. Two more things then. Species distribution. Again, this is a case where you might be given a particular example that you need to work with. So students should be able to evaluate the impact of environmental change on the distribution of species given appropriate information. So they could give you information about any species whatsoever. Please don't come out the exam and say, Miss, you haven't told us about lesser spotted woodpeckers or whatever it is, because they can choose any animal or organism they want. So environmental change affects the distribution of species, including the temperature. So this could cause migration of species, for example. This links into global warming. Yeah. This could be on a daily basis. It says here it could be seasonal, geographic, or caused by human interaction. It could be a daily basis that an organism is moving and responding in terms of temperature. It could be a seasonal thing like migration, or it could be a big shift caused by global warming. The availability of water, again, could be daily or seasonal. And the composition of atmospheric gases. Okay, so the carbon dioxide, the oxygen, etc., that is available in the air, depending on what the organism is. This could be caused by human interaction, either disturbance or the industrial movements that humans are making in certain areas, perhaps increasing the temperature, reducing the availability of water, and completely changing the atmospheric gases. So just be aware that they could give you absolutely any organism, any example, and you need to relate to that. Final thing then, you'll be pleased to hear, is plant hormones. So these are a few extra bits that are for higher tier only. For both foundation and high, you need to know about auxins and phototropism, geotropism. But for higher tier only, you need to know about gibberellins and ethene as well. The gibberellins are the ones that are important in seed germination. So G for germination, G for gibberellins. Ethene controls the cell division and ripening of fruits. And you don't need to know how they work. That's clear there. Look, you don't need to know how they work, but you need to know what they do. Students should be able to describe the effects of some plant hormones and the different ways people use to control plant growth. So it could be that you get a 4 to 6 mark question on different plant hormones, how they're used to grow plants in terms of auxins or ripen plants or for seed germination or whatever it is. Plant growth hormones are used in agriculture and horticulture, so farming and gardening really. Auxins are used as weed killers, we need to know about that, as rooting powders, and promoting growth in tissue culture. That's what you put on the Petri dish when you do a tissue culture cloning. Ethene is used in the food industry for ripening of food, and gibberellins can also be used, especially in garden centres and horticulture, to end seed dormancy, promote flowering, and increase fruit size. So could this possibly be a 4 to 6 marker? Could we be asked about how enzymes are used in how hormones, are used in agriculture and horticulture? That would be quite a really good 6 marker because there's lots of things that you can write about it. So that's it, folks. Separate extra content is available on the Separate Science video that I uploaded earlier. It is a long one, but I'm sure by now you'll just want to skip through to just a couple of things that you need. So all the best for tomorrow. Really do hope the exam goes well, and best of luck.